Hello, students, and welcome to the second part of our unit, the control of eukaryotic gene expression. So we prokaryotic, now we're doing eukaryotic. Let's begin with a prayer. In the name of the Father, Son, Holy Spirit. Hail Mary, full of grace, the Lord is with thee. Blessed art thou among women, and blessed is the fruit of thy womb, Jesus. Holy Mary, Mother of God, pray for us sinners now at the hour of our death. Amen. Okay, so we continue with our gene expression. And the whole point of this kind of presentation is to kind of try to bring it a little bit more together, but also to show you that eukaryotic gene regulation or gene expression is a lot more complicated than prokaryotic gene expression. And kind of what we're getting at here, and that's what this slide kind of explains. Remember, histone proteins are proteins that DNA wraps around. Remember to always read the comments down here when we're going through this. But one thing I want to make sure you understand is the word totipotent, which is right down here. That means that a cell has a full set of genes, and it can become any type of cell. It can become a liver cell, or a skin cell, or an eye cell, or a muscle cell, or anything like that. There are some exceptions in your body, although most cells are totipotent at one point. White blood cells are not. They have different genes in each one, but those are the ones that attack the disease. So totipotent, we'll come back to that word in a minute, but every one of your cells has all of those genes, and essentially this is how do those cells know to make the proteins that make it a skin cell, or to make the proteins that make it a heart cell, even though those cells have all of the genes to make all those points. Or how does the genes in your eye know, the cells know to make a lens behind your cornea? So that's all what gene expression is. How do those cells know how to become the cells that they are? A muscle cell has all of the genes in it to become a kidney cell, but it's a muscle cell. So that's what we're kind of be looking at. This slide talks about how the DNA folds itself up, how it goes from a DNA double helix all the way to a chromosome. You notice sometimes here I put links for YouTube videos that you can go watch that can help. Here are the histones that wrap around. Then there's this one histone called histone 1 that the DNA doesn't wrap around, which helps with the folding. Right here, like this level of folding, and then there's more folding or protein scaffolding. You can look that all up. Here are some vocab words. And what they refer to as your DNA is a lot repetitive. It just repeats. You have the tandemly repetitive DNA, and you have the interspersed repetitive DNA. What I want you to understand is when DNAs make a copy of itself, the lagging strand actually leaves a lot off at the end, and that's called a telomere. And telomerase can restore that repetitive sequence. Telomerase is another enzyme. But that's a lot of the tandemly repetitive DNA, just at the end of a DNA, because it loses a little bit every time the DNA replicates. Now, how can we alter the genome? And when we alter the genome, that is a form of gene regulation. And that's what this is talking about. It actually talks about how gene loss or gene amplification or transposed genes are a form of gene regulation, but they kind of also alter the genome. And that makes certain parts of the body. This slide talks about how a long time ago there was a globin gene, which can help make the hemoglobin, which carries oxygen around in your blood. And then you can get, over time, it evolves into two forms. And it doesn't, it's like we lost the one gene, they're both still there. And then over time, that mutates, and we get all these different forms. And they're all on the DNA. And all these spaces in between, they're called introns, and they can be deleted. And you can form different types of hemoglobin that specifically can do different things. Which is great because different organisms live all over this world with different types of amounts of oxygen and all that. This one here talks about how white blood cells can rearrange these parts, take out different introns to make different things. Here is a video. If you go to this part down here, you can find this video, which helps explain that. Epigenetics, we'll talk about the methylation and acetylation in the next video.